Hi, Ernie.
Hey, good
Nice to
morning.
see you. Thank you for joining. I'm
Mhm.
really grateful that you can take some time out of your day to have this conversation. I I'm very lucky to get to know you. I got to meet you for real at Lab Central, and the work you guys are doing there is impressive and interesting. And I thought it would be a great conversation for the students who are in this program. So maybe we can start there. Could you introduce yourself and say where you work and what you do?
Sure. My name is Eryney Marrogi. I'm a research associate at Dyno Therapeutics. It is, I guess you can say it's an early stage startup. I think mid stage is at this point more appropriate, but I've been working at this company, Dyno Therapeutics, for about a year and a half now at this point in Kendall Square. We are an AAV delivery company. And Yeah, I think that is the highest level overview that I can give about that.
Can you just say what AAV is?
Oh, sure. Yes, it is a adeno-associated virus. It's one of the most popular, if not the most popular, vectors for gene therapy delivery currently in the field.
Right, right. So it's an incredibly interesting and relevant field that you're working in, and it's also fascinating. The whole startup community. I know a lot of people are curious about what it is to work and be part of a startup. So maybe let's start by talking about the science and the company, and then we'll talk a little maybe about the culture of being in a startup. But what is your day like? What attracted you to doing this work?
Yeah, that's a great question. So to start off, I think something that's unique to Dyno is the approach that we take to the problem. So one of the biggest problems that exists in the field for gene therapy or gene therapies that use AAV is they are reliant on sort of naturally occurring virus that will do more or less as it pleases. And oftentimes that means you are using a virus that may not go exactly where you want it to go. And, if we want to treat patients, I think one of the the most important factors is you have a reliable model that you're using. So you know that when you use this virus, it's going to go to the heart and it's going to do this thing. And so one of the biggest difficulties with AAV is that you're using a virus and there are different serotypes of that virus that are maybe better for one thing or another thing. But overall, so far in the field, there hasn't been much luck in getting these serotypes to be more specific than they already are in nature. And what Dyno is doing is applying machine learning to rapidly develop viruses that are able to better adhere to criteria that we want. So for instance, we might take a naturally occurring serotype that's generally really good for the central nervous system and make different modifications to that. So we tend to do mutations on the scale of hundreds of thousands. And we make those mutations in the virus, in the cap of the virus, and then we use animal models to see which mutations had what sort of advantageous effects. So maybe that's increased packaging, sizing, maybe that is higher specificity, maybe that's better binding to a certain organ or tissue type
Mhm.
over another one. And all of that kind of amounts to improved capsids or capsid is kind of like the actual virus itself. It's improved viruses for gene therapy, which
Yeah,
ultimately means better gene therapy for patients.
yeah, yeah.
Yeah,
It's
and so
fascinating. It's a platform, right, that you're developing. It's not that you have a particular gene therapy target that you're working towards. It's really a platform technology that you are focused on. And, in that sense, I see it very engineering focused, right, as well as scientifically, life science focused, which is very cool. And it's just so interesting to hear, you mentioned as you describe the science of it, right, and then the machine learning of it, and all the testing of it. So, which aspects of the platform development are you most focused on?
I have worked since I've started Dyno, since Dyno's kind of infancy, on everything that is downstream of the machine learning. And the loop, Dyno's platform is kind of more of like a feedback loop where you have machine learning that's kind of starting step one, making the designing the library or designing the viruses. And then you have step two, which is making the viruses, and then step three, which is testing the viruses. And then that testing phase then loops back into the machine learning to then generate the next iteration of the library. I have worked on, I guess, the middle and the last stage, which is the generating viruses, and then the testing phase.
Yeah. We talk all the time in BioBuilder about that design, build, test cycle as an engineering cycle. And it's it's really just amazing to hear it so directly applied to such a vital question and a growing company. So, so awesome. So your training must be quite broad, and I want to ask you how you got to where you are, but just backing up just to ask before I forget about, how do you like working at a early stage and now mid-stage startup?
So I joined, when I joined Dyno, we were not very big. It was, I think I was maybe like the 8th full-time employee. I don't really remember at this point, but now we're closer to 40. So the changes that I've seen in the company have been pretty stark from when I started. When you kind of start at an early stage company, you wear a lot of different hats that don't necessarily apply to your job title. That might include sometimes going to business meetings, even though you have no business training. But now we have like a really great team for that sort of thing. It was spending a lot of time doing company culture work. That was spending a lot of time on just generating the life of a company. And now I think my focus is a little bit more finite to the science. So that transition has been exacerbated, I think, by the rapid growth of the company, but it's one that happens for everybody, regardless of whether you're at a biotech startup or a regular tech startup or just a small company in general that grows. And it can be difficult to lose out on some of the diversity of what you do, but I think ultimately when you get back to like what you're good at, everybody is happier in the long run.
Yeah. Just as an aside, starting up BioBuilder, it's a nonprofit organization, but it is also a startup, and I wore many hats, from curriculum designer to chief bottle washer to finance and communications and all that. And yes, as you grow, you start to sort of lean back into the things that you're trained to do, and you find experts in the areas that you still need help in for the company. So
exactly, yeah. I mean, it's great. It's great because you get a little bit of everything. And you wouldn't really get that anywhere else. But ultimately, like I said, I think everybody is happier in the long run when you don't need to wear a lot of different hats and different things every day.
Yes, fall back on the things that you're like, oh yeah, I know how to do that. Right. So then actually maybe that's a good segue to ask you about your training and, when did you know you were interested in doing science? Where did you go to school and what did you study?
Sure. I think that I've had a little bit. I've had an easier time with the scientific route just because of the path that I took. So when I was in high school, I grew up in Virginia. I did a summer program at the NCI, the National Cancer Institute in Bethesda, Maryland, and I really enjoyed it. I found the work to be really fresh for me. It kind of tied what I was learning in the classroom with the real world, kind of like you just mentioned, right? With the process of designing, you learn about that in the classroom, but sometimes you wonder, like, what does that look like in real life? Well, that for me was that sort of summer experience. That looped into a more continuous role there, working in the lab. And I also did the Intel Science Fair. I don't know if that's still around anymore, but I did that all 4 years of high school. And I very much enjoyed that. I strung that into a undergrad degree at Virginia Tech in biochemistry.
Mhm.
And naturally, biochemistry is very science oriented, entirely so. And during that time, I was working in a protein modeling lab. So I did a lot of computational work. Um, During my summers, though, I actually spent them in Boston in the Church Lab,
Mhm.
George Church Lab at Harvard Medical School. And I was working at the time on something completely new for me. After spending several years in cancer biology, I now was working in mosquito engineering, vector engineering. And that was a completely new experience. You know, you learn a lot of things that are unintuitive, and I learned a lot about the mosquito that I would not have ever learned beforehand. I spent 2 summers working on the Gene Drive project, as it's called, in George's lab. And then when I graduated from college in 2018, I stayed on in the Flaminia Catarrhea lab, and she was working with George on the mosquito project at Harvard School of Public Health. I worked on that after graduating through her lab.
Mhm.
And I then joined Dyno after a year there. Eric, our CEO, was actively recruiting from George's lab at the time, and I was ready to, I think, move away from academia and into industry, and the opportunity kind of just aligned with what I
Yeah.
was looking for.
Yeah. Well, it sounds like, again, a broad set of skills and training that didn't have a direct line to the things that you're doing now. But, when you learn how to ask good questions and answer good questions, that carries over from, mosquitoes into adenovirus to anything,
Yeah.
is the ways of thinking and the ways of being and doing. Yeah.
Yeah, I guess I like to think of it as there are sort of hard skills that you learn, which are maybe like PCR. I don't know if
Mhm.
this is something that a lot of the students are aware of, but there are certain lab skills that you learn. You learn how to run a gel, you learn how to do Western blotting, you learn how to do PCR, you learn how to do all these things. And these hard skills, maybe they need tweaks between whatever work you're doing, but they're pretty translational. And then you have your soft skills, which are, as you're mentioning, the ways of thinking about ideas and thinking about problems and coming up with solutions. Those skills are equally transferable, and I think oftentimes more so, just because you can kind of plug and play the thought process of, yeah, I mean, like the details around mosquito engineering are definitely different than AAV, but the principles of using tools are the same, and so
Right.
your thought process
Yeah.
ultimately stays the same.
Yep, yep. A good experiment is a good experiment, and data analysis requires sort of a skepticism about what you're looking at and seeing, and
Mhm.
that's true no matter what you're actually studying and doing. So, yeah, I think that's great. The other thing that I really love about your story is um, the notion that it was an early experience, a high school experience that sort of directed you towards the application of the things that you're learning in classes. Because I do think that classes do teach a lot of the good science classes are teaching those hard skills, how do you run a PCR and things like that. But I think the realization of how it all fits into the actual being of a scientist and the doing of science is really most clear when you have a chance to be in a lab, especially, and that should come earlier. I'm all about getting that sort of experience of being a scientist or an engineer early, giving people early opportunities to do that.
To me, I think the earlier that someone has an exposure to those hard skills, the faster they can develop the softer skills, because you
Mhm.
no longer require an awareness about um, how do I do a qPCR, how
Right.
do I do a sequencing run. That
Mhm.
sort of becomes second nature, and you can
Mhm.
just focus on how do I use those things to my advantage. It's kind
Yeah.
of like playing an instrument.
Right.
First, you learn the scales.
Mhm.
And then once you know your scales super well, then you can just do anything. You don't
Yeah.
need to think about it.
Yeah.
And it's all about just developing the muscle memory. And
Yeah.
all of a sudden, you no longer one day have you no longer have an aversion to doing a complicated experiment just because it's, you know, it's
Right.
everything to you.
Your technique is there, you know, the foundations are there, you just are growing from it. Yeah, yeah. I think it does make you brave. I completely agree. I think that these experiences and the confidence that you have in the sort of base technique, the scales of doing science, which I love that analogy, I think is great. And it does give you confidence to do more. And that's what we need, right? We need young people coming into this who are not afraid and who are bringing their good ideas and able to do more. So that's awesome. Now, I've been fortunate not only to know you a little bit through Dyno and the connection we have through Lab Central, where the Learning Lab is, but also through BioBuilder, where you have been a mentor to teams and students. So do you want to say anything about your interest in being part of BioBuilder or what you've done as part of BioBuilder?
Sure. Yeah. As I mentioned, I worked on different projects through science fairs when I was in high school. I always valued that early exposure. And when I was in college, I tried to still be involved with that process as a judge at those science fairs. And as I have graduated and kind of moved on, I was interested in exploring like what does that look like now for me. And luckily, I think I saw an email that had been circulated at Harvard when I was there, and it seemed fitting for that sort of experience that I was looking for. And I'm glad I reached out at the time. That's over 2 years now that I think about it. So it's been a while, and I've worked with different teams, but I've also worked consistently within the same two schools, which I think it's been easier for me because I've been able to develop a connection with the teachers and then certain students, certain faces you see from year to year. But I think the best part of that experience has just been being able to see the ideas that these students are coming up with that are they're out there, you know, they're definitely out there, and it's really great to see that they're not yet grounded on the reality of like how difficult it is to do certain things, and that comes with time. But I think it's easier to learn that than to learn how to think very abstractly and creatively.
Right.
So the projects that I've seen have been, I think, really neat. They've been really cool,
Yeah.
and the work that the students put in, it's awesome to see. They clearly have a lot of passion about it. I imagine that most of them are not just doing it because they have to, and that's always a good thing as
Right.
well.
Right. Yep. Yep. Well, I've heard great things about your mentorship of the teams, and that is, you know, what you articulated is exactly the sort of ethos that we try to make sure gets conveyed here through BioBuilder, is that, you know, your good ideas are what really matter, and that we will teach you the techniques and the tools for you know, testing them and you know how you might build them, but that ideas really do matter, and that people, it's sort of surprising, at least, you know, when you learn science, especially in high school, it's sort of a big collection of facts, right? It's like everything that's in a book. And so it doesn't always seem clear that there's actually a lot to do with it, right? It's not just passive sort of learning of lots of things already known, but it's actually the way you put it together, the way you can actually, you know, craft it into a technology and a solution that you want to design, right?
Exactly. Yeah.
Well, it's wonderful. So, is there anything, last question that I'll ask you, and then I'll let you get back to your day. Is there anything that you kind of wish you knew way back when that you know now and think is important to keep in mind, or something that
Yeah. I you know, I think every experience that you have teaches you something. You know, some experiences teach you more than others. But what something that I would always, I think I still recommend to people who ask me this sort of question is to think about what an experience can offer you in terms of where you would like to be. If your eventual goal is, for instance, if you really want to be a physician, think about what sort of experiences will get you to be like the best version of that goal.
Right.
And it can be really easy, I think, to chase prestige, and very easy to chase, very attractive options that don't really fit well with your story.
Right.
And the sooner you can get over that desire to want that sort of stuff, I think the easier it becomes to be really happy with what you're doing.
Yes.
So that's my piece of advice, is to think very critically about like what is something offering me in terms of where I want to go, and does that help me not only get there, but once I am there, does it help me to be the best version of that, whatever it is.
I think that's awesome. To continue to try to be the best version of ourselves is going to be a very key element to success and to happiness. I think that's exactly right. Exactly right. Well, I wish you certainly great success and great happiness. And I appreciate your taking the time to just kind of articulate and walk through your story a little bit.
Absolutely. Thank you very much for speaking with me. And I am always available for questions. So.
Awesome. I will let folks know. Thanks.